is biotechnology? Biotechnology is the use of organisms to produce useful products. For thousands of years, humankind has used biotechnology in agriculture, food production, and medicine. The term itself is largely believed to have been coined in 1919 by Hungarian engineer Károly Eréki. Restriction enzyme. A restriction enzyme or restriction endonuclease is an enzyme that cuts DNA at or near specific recognition nucleotide sequences known as restriction sites. Swiss microbiologist Werner Haber was one of the recipients of the 1978 Nobel Prize in Physiology, an award he earned for the discovery of restriction enzymes. Starting to use restriction enzymes, the first major application was as a tool for cutting DNA into fragments in ways that would make it easier to study, identify, and characterize genes. A second major use was as a device for recombining or joining DNA molecules from different genomes, usually with the goal of identifying and characterizing a gene or studying gene expression and regulation. Recombinant DNA technology using restriction enzymes. Steps for recombinant DNA technology. Step 1. Plasmid DNA and DNA from another organism are cut by the same restriction enzyme. This produces molecules with complementary single-stranded ends. Step 2. The two types of molecules are mixed so that their sticky ends pair. DNA ligase then forms covalent bonds at junctions linking fragments. Step 3. The recombinant DNA molecule is then transformed into a host cell, such as bacteria, where it is copied and turned on to produce a gene product. The biotechnology revolution. Gene cloning and PCR allow scientists to make a large amount of DNA from only a small fragment. But how do these technologies work? Using reverse transcriptase to clone expressed genes and the polymerase chain reaction, making DNA from RNA. Cloning involves the creation of DNA from mRNA and represents a reversal of the central dogma. The reversal of the central dogma concerns the making of DNA from RNA. It is the process by which DNA is transcribed to RNA and RNA is then translated to proteins. Now when we go back from RNA to DNA, that is called reverse transcription. Discovering reverse transcription. Howard Temin hypothesized the existence of an enzyme capable of making DNA from RNA as early as 1964. The central dogma reversed in 1970. How PCR works. The following are essential chemical ingredients needed: DNA polymerase, a small amount of DNA to serve as the initial template, 3 deoxyribonucleotides, a few necessary ions and salts, a pair of primers with exposed 3' OH groups that will bind to the particular sequence of interest in the DNA template. The polymerase chain reaction. 1. DNA is heated to 90 degrees to 100 degrees Celsius to separate the two strands. 2. The DNA is quickly cooled to 30 degrees to 65 degrees Celsius to allow short single-strand primers to anneal to their complementary sequences. 3. The solution is heated to 60 to 70 degrees Celsius. DNA polymerase synthesizes new DNA strands, creating 2 new double-stranded DNA molecules. Each time the cycle is repeated, the amount of target DNA doubles. Advances in PCR technology. As with all technologies, scientists continue to improve both PCR and the cloning process, thereby ensuring that these methods will play a role in genetic breakthroughs for years to come.
QTLs is known as quantitative trait loci and involves creating what is known as a mapping population. They are chromosomal segments that carry genes. QTLs can be used to identify important genes that can be genetically engineered into crops. Although it may take years to do, it has been implemented in tomatoes. Scientists have found 28 QTLs for tomatoes, as it took 10 years to be localized. One of these QTLs FW 2.2 to a narrow chromosomal region in order to clone it. Due to this, they found that FW2.2 QTL corresponds to the ORFX gene. It was found that injecting these transgenes into a cultivated tomato reduces the size of the tomato. So, by selecting tomatoes without FW2.2 QTL, farmers are able to better breed their tomatoes. QTLs are not just used for agricultural purposes. Besides plants, scientists are looking for ways that QTLs can answer questions about the quantitative genetic basis of evolution as introduced by Carl Pearson and Ronald Fisher during the early 1900s. Genetically modified organism is one that can have been altered through recombinant DNA technology, which involves either the combine of DNA from different genomes or insertion of foreign DNA into a genome. For example, the goldfish is the only recombinant DNA approved for human use by the U.S. Food and Drug Administration.
Gene therapy is an experimental technique that involves the transfer of therapeutic genes into specific cells of patients in order to repair defective ones. The most common method of delivering therapeutic genes into target cells employs using a virus vector. Viruses introduce their genetic material into a host cell as part of their replication cycle. Scientists can capitalize on this process by removing the disease-causing portions of the viral genome and adding foreign therapeutic genes. Now, instead of producing viral toxins and causing disease, the cell infected by this virus will be able to produce the protein or enzyme it lacks. The vector can be injected or given intravenously, or a sample of the patient's cells can be removed, exposed to a vector in a laboratory setting, and then the cells containing the vector are returned to the patient, and the altered cells can then produce the desired protein. Jesse Gelsinger's death in 1999 created a setback for the field of gene therapy. He suffered from ornithine carbamylase deficiency, which is an inability to break down ammonia. 4 days after receiving an injection of adenovirus carrying a corrected version of the OTC gene, he died due to multiple organ failure. In the aftermath of this tragedy, government and scientific inquiries were initiated. Investigators learned that clinical trial scientists had not reported the adverse reactions to gene therapy, nor did they explain the serious side effects from the animal studies to the patients during informed consent discussions. Thus, the US FDA scrutinized gene therapy trials across the country, halting and shutting down several of the programs. The question remains whether gene therapy can ever recover from the setbacks and keep its promise as a cure for genetic diseases. According to the World Anti-Doping Agency, gene doping is the non-therapeutic use of cells, genes, genetic elements, or of the modulation of gene expression having the capacity to improve athletic performance. The science behind gene doping. Now, H. Lee Sweeney, a professor of physiology and one of the leading researchers in the field of gene therapy, has garnered attention because of his discovery of a way to potentially reverse muscle degeneration caused by diseases. Sweeney and his colleagues observed that when a protein called insulin, like growth factor 1, interacted with the cells on the outside of the muscle fibers, it caused the cells to grow. The primary focus from a gene doping perspective is its ability to increase the rate of cell growth, in particular muscle cells. The field of gene therapy, and by extension gene doping, is full of unpredictable and dangerous results, which is why Sweeney, Evans, and other researchers who have identified DNA targets that could potentially be exploited for gene doping are the first to emphasize that the research is still at only an experimental stage and further research is required.
Testing for traits unrelated to disease. Diagnostic testing supplies the technical ability to test pre-symptomatic at-risk individuals or carriers to determine whether they will develop a specific condition. It is an attractive choice for individuals who are at risk for diseases, as well as people who might carry genes that have genetic reproductive recurrence risks. With advances in single-cell diagnostics and fertilization, technology embryos can now be created in vitro. Then embryos that are not affected by a specific genetic illness can be selected and implanted in a woman's uterus.
According to social science research, genetic testing may and may not harm the at-risk individual by creating undue stress and anxiety. Currently, available tests include those for eye color, handedness, active behavior, nutritional background, and athleticism. The results of the study may be surprising to many people, as one major concern associated with testing for non-disease traits is the fear that those people who do not possess the genes for a positive trait may develop a negative self-image and/or inferiority complex. Another matter bioethicists often consider is that people may consider that they carry some genes associated with physiological or behavioral traits that are frequently perceived as negative. Moreover, many critics fear that the prevalence of these traits in certain ethnic populations can lead to prejudice and other societal problems. Creating designer babies. Trait selection and enhancement in embryos raises moral issues involving both individuals and society. If parents are allowed to manipulate the genes of their children to select for certain traits when the children themselves cannot give consent, suppose a mother and father select an embryo based on its supposed genetic predisposition to musicality, but the child grows up to dislike music, will this alter the way the child feels about his parents and vice versa? In terms of society, it is not feasible for everyone to have access to this type of expensive technology. Only the privileged members of society will be able to have designer children. That poses greater intelligence or physical attractiveness.